I'm David Hay. I'm a Professor of Tissue Engineering at the University of Edinburgh's Centre for Regenerative Medicine. So our focus, we use pluripotent stem cells to generate human liver tissue. We use that to model humans in a dish, and we also use that to generate implantable liver tissue for clinical work. We've used a number of Promega's assays to understand if we're creating healthy liver tissue, to understand if that liver tissue is functional, and also if that liver tissue can be used for our experiments.
I am Alvile Kasarinaite, the PhD student under the Precision Medicine MRC grant, and I'm working with Professor David Hay in Centre for Regenerative Medicine. I'm modeling sex-dependent differences in non-alcoholic fatty liver disease using stem cell-derived liver tissue. And I'm working on 2D liver different cell types and then maintaining the culture in 3D. We're using a lot of Promega assays in general in our lab, but mostly we were focusing on cytochrome P450, different enzymes and their screening assays, the real-time Glo MT viability assay, and glycerol and triglyceride assays. So previously in our lab, my colleagues used the red oil O staining to measure the content of lipids within the liver spheres. However, it's a very laborious procedure, so we were looking for something that is quick, quantitative, and could be used for multiplexing. And we found this Promega assay of Triglyceride-Glo. We're using Triglyceride-Glo for our liver spheres. So we take the cell lysate, the sphere lysate, and then we measure the triglyceride content within our spheres using biological and technical replicates. Usually the whole assay takes around 2-3 hours to complete, and I'm starting in the morning with the harvesting of the liver spheres and the preparation of reagents, and since there are some incubation times, I can do other lab work or relax and have lunch. We can indeed see that our liver spheres are accumulating the triglycerides, and we are further optimizing and testing on different conditions. So we're trying to see whether our liver model can indeed incorporate the triglycerides and whether it accumulates them. And also we would like to see in the future whether there is a difference between the steatoses and our control samples. NAFLD is characterized by an excessive accumulation of triglycerides, so we're hoping to show that our liver sphere model is able to recapitulate this difference within our healthy and the steatose sample.
So the bigger picture is trying to find new medicines to treat disease in humans by modelling them in the dish, and also to generate plantable liver tissue that could be used within the clinic to help patients with failing liver function.